Format for Lab 3 Histology is that we are going to go over the four main types of tissue. We are going to spend a lot of time on epithelium and connective tissue because these are the hardest concepts for students to understand. So if you look at your attendance sheet for Lab 3, it has a couple tables that you want to fill out as you're going through the PowerPoints and the videos. The main things I'm going to ask you to fill out are going to help you differentiate between the types of epithelium. So paying attention to things like shape, numbers of layers, where you would find the nuclei, as well as function and location. These are questions that I want you to be asking yourselves as you practice with the microscopes and with the images online, because these are the questions that will help you answer lab exam questions. The same thing applies to the next video on connective tissue. So the second table you fill out will ask specifically about fibrous connective tissue because that is probably the most confusing type for students to grasp. So this is all designed as practice to get you used to identifying those types so that when you get to the lab exam, you're not totally lost. And then on page 3 of the attendance sheet, there's just a couple questions about muscle and neural tissue. So these tend to be easier to identify. They look a little more distinctive, but they're still important to know. So for this lab and for studying for the Lab Exam 1, Canvas modules are going to be your best friend. So under Lab 3 Histology, we have tons and tons of examples of epithelium. It's all organized by type. And then we have connective tissue. So if you're really struggling with fibrous connective tissue after today, look at these images. Go through them. This is exactly what you'd see under a microscope. So practice identifying these types of tissue and the components that we'll talk about over the next couple of minutes. Same thing for supportive connective tissue. And then we have just one example of fluid connective tissue, a blood smear. So most of today will be spent sorting through epithelium and connective tissue. So let's dive right in, starting with epithelium. So big picture stuff, this is going to come back over and over again as we look at organ systems because epithelium is going to coat linings anywhere that the body is exposed to an external environment. So we'll see this pop up when we talk about blood vessels, the digestive tract, the skin system. We're going to find epithelium all over. So if you can start to recognize the types of epithelium now, that's just going to help you on our later labs. We also need to bring back the concept of a basement membrane. So we talked a little bit about that in Lab 2. You had one exercise asking you to identify a basement membrane off of a slide. So we are going to see tons and tons of those today. A great example, if we go to the Human Protein Atlas, we find basement membranes, for example, in the respiratory system. So if you put a slide under a microscope and you're at about 4x, this is what you would typically see. We're going to zoom in now because we want to hone in on the epithelium here. And this starts to look pretty familiar. So we have a layer of cells forming that kind of first floor of the house analogy that we used Friday. This is your epithelium, and it's sitting on top of a basement membrane, which is made of connective tissue. So this is not exclusive to the respiratory tract. We'll see it all over as we work through future units. All right, so how do we identify different types of epithelium? Well, epithelium is largely a cellular tissue. So we're going to be dealing mostly with cells. And then the other component is called extracellular matrix or ground substance. It's sometimes abbreviated as ECM. And really all that is, it's water and a lot of large molecules like proteins and carbohydrates, things that are going to enter cells and help nourish the cellular material. But we're mostly focused on cells right now, so we're not going to worry too much about that matrix. We're going to see epithelium in membranes, lumen of blood vessels, any sort of internal passage, and we'll also see them in glands. So as you're filling out that table today, watch out for the example locations that I give in the PowerPoints and in the videos. Another thing to note about epithelium is that it is avascular, meaning it has no blood supply, but it is highly innervated. And we'll talk about that more when we bring in the integument or the skin system next lab. We'll see how we can tell the difference between the superficial layer of the skin and the deeper layer based on those characteristics. So structurally, we already covered the basement membrane, so I'm not going to talk too much more about it today, but it's a great example of how epithelium and connective tissue are going to interact. So one big concept to know as we work through future units is we're not going to see just one type of tissue, and the respiratory tract is a great example. We are dealing with connective tissue and epithelium, and you'll need to know the difference on a slide so that you can navigate the organ systems that we look at. So these basement membranes are going to be really helpful for that, but we'll continue to work through that and have that big concept be a major theme with all the histology that we look at. All right, so ways that we can distinguish between types of epithelium are going to be by shape and the number of layers. And we'll also see some specialized organelles such as cilia and microvilli that might give us some clues about where in the body we are seeing this epithelium. So let's start with kind of the simple version. Simple means one layer, so we're going to have 4 types of simple epithelium. Simple squamous is going to be a single layer of flattened cells. And the way I remember this is I think squamous sounds a lot like squashed, and so these cells look flattened or squashed. Now, we find this simple squamous epithelium anywhere that we have a lot of exchange of material. So having one layer of cells to filter through versus multiple layers is going to make secretion, absorption, any sort of exchange a lot easier between organ systems. So for example, we would find these in the air sacs of lungs. We'll find them in blood vessels. We might find them in the kidneys as well, anywhere that there's a high level of exchange of materials in the body. So next tissue is simple cuboidal. Based on the name, we know it's a simple single layer, and then the shape of the cell is going to be roughly cube-shaped or cuboidal. So this will not always be perfectly cube-shaped on your slides. So you want to look at kind of the length to width ratio here. It's roughly equal versus where the squamous epithelium is really flattened. They're much longer than they are tall. So cuboidal, much more round or cube-shaped. Cuboidal cells will be found anywhere that we have a lot of absorption or secretion. We'll find them a lot in glands, actually. So almost always a glandular tissue will have either cuboidal or columnar epithelium. So we actually have a kidney slide we'll look at, which is, pretty much all cuboidal epithelial cells. Other places we might find them would be like the lower respiratory tract, and then in a lot of different types of glands around the body, including some that we'll look at in the skin system next lab. Next up, we have simple columnar. So again, it's one layer. The shape is column-shaped or columnar. So these guys are going to be much taller than they are wide, forming basically a row of columns. So these are again going to be involved in absorption and secretion. So we'll find cuboidal and columnar in some overlapping locations around the body. One place being the digestive tract. So from the stomach to the anus, we'll talk about columnar epithelium. Uterine tubes and some parts of the kidneys where we have a lot of absorption, we'll also find this type of epithelium. Now this last one is tricky. Pseudostratified columnar epithelium doesn't really look like these other tissues. It looks like a stack upon stack upon stack of cells. So this is where it can get tricky for students. If we followed one cell of this pseudostratified epithelium, we would find that each cell is actually anchored to that basement membrane. So their appearance is deceptive. We have nuclei at different heights here, which creates this appearance of multiple layers. But in reality, this is a single layer epithelium attached to that basement membrane. So this is found in one specific area of the body, which would be the respiratory tract, specifically the upper respiratory tract. So let's go back to that nasopharynx example. This looks a lot like pseudostratified columnar epithelium. And there's one additional feature here, the cilia, that helps us know this is the upper respiratory tract. So cilia are actually involved in moving things along the respiratory tract in what's called a mucous membrane. So we'll look at that a little more on the Yale site. You can actually pull it up here. So here's another perspective of that pseudostratified columnar epithelium with the cilia. So the cilia, if you remember from last lab, have a motile function. They can actually beat in rhythm with one another and help propel substances along the respiratory tract. So things like mucus or particles that go down the wrong way, it's going to help remove them from the airways. So a lot of times us instructors like to ask about pseudostratified ciliated columnar epithelium. So just keep an eye on this tissue. Make sure you look at our slides available in the modules because this tends to come up a lot on lab exams. So that is all simple. And then we have stratified counterparts. So we have basically stratified squamous tissue, stratified cuboidal, and columnar. So they're going to be exactly the same shape of cells, but now we're dealing with multiple layers or strata. So first off, we have stratified squamous epithelium. This is going to be found in the skin or at the epidermis, that superficial layer of the skin. And anytime we have stratified layers, that's going to have a protective element. In this case, stratified squamous epithelium is going to create a nice barrier between our external environment and the underlying tissue. If you remember, this tissue is going to be vascularized and innervated. The epithelium is also innervated, so we want a lot of barriers between the external environment and those more sensitive receptors. So stratified squamous epithelium is also going to protect from any sort of mechanical stress. So if you think about if you scrape your arm against something, layers of dead cells can actually come off. But if you just scrape yourself lightly, you're not really going to feel it because just the dead cells are sloughing off. Now, if you get a worse scrape where maybe you're drawing blood, at that point you have penetrated past the squamous epithelium, and that's when it starts to hurt. But for small mechanical stresses, this is a protective layer. It also prevents water loss. So we can, if water is evaporating from the skin, we can actually get dehydrated very quickly. So if someone is sweating a lot and they're not replacing fluids, that's when it becomes dangerous and they get dehydrated. Well, having these multiple layers is going to help prevent that water loss. So anytime you hear stratified squamous epithelium, think epidermis or skin. Next up, we have that stratified cuboidal epithelium. So this tissue is largely going to be found in ducts. So we'll talk about the sweat glands next lab as well because we have sweat glands in our skin system. And so they're going to be involved in absorption and secretion. So let's see if we can find an example on the Yale histology site of stratified cuboidal. Okay, I'm not seeing an example here, so maybe we can pop over to our modules. We're looking for stratified cuboidal. Okay, it doesn't look like we have an example here. So I will make sure to point that out in class, and I'll go ahead and add an example to your modules because I definitely want you to be able to identify this type of tissue in ducts. Okay, so next example is stratified columnar. This, again, it's a columnar shape, and we're dealing with 2 or more layers. And make sure you don't get this one confused with pseudostratified up top. So again, we have to look to see if each cell is attached to that basement membrane, or are we dealing with layers of cells. So if we look closely here, you can see there's 2 layers of cells, and the top layer does not connect to the underlying basement membrane. So that's how we know it's stratified columnar versus pseudostratified. So similarly, This is going to be involved in absorption and secretion. We're dealing with more layers, so it's also going to have that protective function. So you should hopefully be noticing a pattern between these types of tissues and where we would expect to find them in the body based on their shape and numbers of layers. Last type is pretty weird, actually. This is transitional epithelium, and it's found in just a couple places in the body, in our urinary bladder, ureter, and urethra, so all urinary system. So this is multiple layers of cells, but it gets interesting because these cells will change shape based on how they are stretching. So when your bladder is really full, these cells are actually going to stretch out and appear a lot more squamous or flattened in shape. And when we empty our bladder, these cells will revert back to this more regular position. They're more cuboidal right now because they're not distended. But this is useful because it means when we drink water or other fluids and form ultrafiltrate in urine, it means we don't have to immediately go to the restroom. We can hold it a little bit while the bladder fills up. So I know we do have an example of transitional epithelium. Let me find it here. So this is the ureter of a cat. Kind of fun. And the transitional epithelium is going to be this layer of cells here. So I guess the best way to look at this is right now the cells appear cuboidal in shape, but as this flattens out, we're going to see maybe more along the edges squamous epithelium or squamous shaped cells. Let's see if our other example here shows, okay, this one shows a relaxed bladder. So in this case, the bladder is not full. So this is a wider scale view here. So right now, the bladder is not distended, so you might see a lot more of these folds on your slide. So that's one way to kind of tell. There will be some variation in the slides we look at, so you might have a distended bladder where the tissue appears more squamous, but we'll definitely look at that more in class, just because I want you to get a lot of practice with it. And let's see if Yale has a transitional one. Okay, so if this isn't confusing, then I definitely recommend the Yale site. They have most of the tissue we've talked about, more so the simple than the stratified examples, but they do have some nice transitional epithelium. So this one, it's a little more clear where the basement membrane is. So this is going to be our external apical surface, and then deeper we have the basement membrane. And you can tell this is the basement membrane based on the difference in color. It's a little lighter on the right side because this is connective tissue. On the left side, we have our epithelium. So one way that will help with looking at transitional epithelium is you can also look at the nucleus placement. And I understand this can look a lot like pseudostratified columnar epithelium. But if you look closely here, you can see multiple stacks of cells indicating that it is a stratified tissue. And another thing to note is when I'm testing you on these things, on these concepts, pay attention to clues in the questions. So if I ask you, identify this type of tissue commonly found in ducks, well, we know that cuboidal epithelium is commonly found in ducks. So that narrows your choices now. We could be talking about simple cuboidal or stratified cuboidal, but that narrows your choices down from all these other types of epithelium. So let's use transitional epithelium as an example. If I were to ask you this, I know that it can easily be confused for these other types of tissue, so I might clue you in by saying, identify this type of tissue that is commonly found in the ureters. Well, if you know the location of transitional epithelium, then that definitely narrows down your choices as well. So that's part of the reason we're filling out the table in this worksheet to help give you some context for these types of tissue. So that is it for this video. Next up will be connective tissue, but I highly recommend checking out Yale's histology site. If you're looking for slides, you just need to scroll down a little bit and then you can click on all of these links here. And then we have tons and tons of examples listed here. I'll make sure to add a stratified cuboidal so you can have an example of that as well.